So we're going to talk to you about the work that we've been doing to understand the molecular basis of hereditary spastic paraplegia. So I think we've met a few of you before, actually, either in person or over Zoom. But just to give a bit of background, so my name's Emma Baple. I'm a clinical genetics consultant in Exeter, and I also work with Andrew in the university. One of the focuses of our work is hereditary spastic paraplegia and other motor neuron degenerative disorders, which we'll talk to you about today. But I also am the medical director for our diagnostic lab in the southwest, so Exeter and Bristol, and we do quite a bit of genetic testing for hereditary spastic paraplegia. So we're going to talk to you about all elements of that today. So
I'll introduce myself as well. So I'm Andrew Crosby. I'm Professor of Human Genetics in the medical school, as Emma mentioned. My role is simpler than Emma's. I undertake research into genetic conditions, obviously particularly HSP. Sorry, Emma, I interrupted. Please carry on.
That's all right. That's all right. So I'm not going to tell you about what hereditary spastic paraplegia is, because we're not here to give you a lesson on something you more than likely know a lot more about than we do. But we did want to talk to you a little bit about why the work that we do on other motor neuron degenerative disorders and on motor neuron degeneration is relevant to HSP and not just the work that we do on HSP. And that's because HSP belongs to a wider group of disorders that affect the motor neurons. And we have the upper motor neurons, which are the motor neurons primarily affected in all forms of hereditary spastic paraplegia. But we also have our lower motor neurons. And when we look at motor neurons and the processes that control them in health and disease, it helps us to understand not only HSP, but other disorders where those processes malfunction and we get degeneration of those particular cells. For those reasons, we choose to look at not just HSP, but the other wider group of disorders that we know overlap both clinically and genetically. And Andrew will tell you a little bit more about that in a moment. It's because everything that we learn helps us to understand the wider role of those cells and the wider role of the processes that control those cells in the body. So Andrew and I, as Andrew is explaining, are both geneticists. That's our bread and butter is genetics. And I suspect all of you have come across a geneticist at some point in your journey. But genes are essentially the code that control everything about us. We've got about 20, well, people argue about this actually, we've got somewhere between 22 and 25,000 genes, depending on which geneticist you ask. And we only understand what about 7,000 of those genes do. So we're only about a third of the way there in terms of understanding what those genes are doing. We suspect that every single gene in our genome will cause a human disease of some sort. So we've still got an awful lot of genes to determine, not just for HSP, but for every human disease. Genes are the code, essentially, the kind of blueprint for life. But genes make proteins, so they're the code that allow our body to make proteins, and proteins are the building blocks of life. So the genes are essentially the code that control everything about us, the way that we look and the way that our body works and functions and develops. What we do initially is try to identify or discover the gene that causes a particular genetic condition. So we have discovered 16 genes that are associated with hereditary spastic paraplegia, different forms of hereditary spastic paraplegia. And we are still discovering new genes every day. Now, that is useful information to patients who have that condition, because we can say, we tested you, we know what gene has caused your condition, we know somewhat about what other patients who have changes in that gene suffer from in terms of what medical problems that they might have, and we can also offer other members of the family a test if that's appropriate to do. But in terms of what that means for patients who have that condition themselves and their own treatment going forward, identifying the gene is really only the first step. We want to go on and understand what that gene does in the body. And in the case of HSP, what does that gene do in motor neurons? What messages, what proteins is it making, telling the body to make, and what are those proteins doing in terms of the function of a motor neuron? Are they important for the way that the motor neuron grows and develops? What are they important for? Because by doing that, we have the potential to say, well, okay, we know what's going wrong when that gene doesn't work properly. Is there something that we can do to intervene in that process? We know what it's meant to do in a person who's healthy, and we know what's going wrong in a person who has a particular condition. What can we do to intervene in that process? So that's just the first step, identifying the genetic cause. We then want to go on and learn about what is happening inside the cells in the body. But as a geneticist, I'm obviously going to come back and tell you that genetics is important, because I believe it is. It is important, and it does give important information to families. So in England, the NHS launched something called the Genomic Medicine Service in 2019, and that was to enable us to standardise genetic testing across England and to best use new technologies that allow us to do genetic testing at scale and to look at lots of genes in people who have a condition that might be caused by lots of genes, so conditions like HSP. That service is, I think, extremely exciting because there's no other service like that in the world. We are now able to use a technology called whole genome sequencing, which is a technology that allows us to look at all of a person's genetic information, all of those 22,000 genes and everything that's in between. And we can do that in a single test, and I can get the results back from that sequencing actually in a matter of weeks. It takes a bit longer for us to interpret the results of it, but in just a few days, a few weeks, we can get all of that genetic information. And the way that we now test for hereditary spastic paraplegia is using that technology, whole genome sequencing, in both adults and children. We want to be able to do it at scale, and we want to be able to test lots of genes in parallel because it gives answers quickly, but it also provides the opportunity for discovering new genetic causes of disease and for looking for new treatments for genetic disease. So in terms of the gene panels that we can now look at for hereditary spastic paraplegia, we look at 76 genes in parallel through whole genome sequencing in adults and 70 in children. Coming back to what I was saying to you previously about the fact that we, when we think about one motor neuron degenerative disorder, that information is applicable to other motor neuron degenerative disorders, that's actually influenced the way that we do genetic testing. So we know that patients who have some conditions that are genetically variable, so different genes can cause them, can overlap in their clinical presentation with other genetic conditions that are similar. So, for example, we know that some patients with hereditary spastic paraplegia might also have other features like problems with their balance that mean that you might want to look at additional genes that cause that group of conditions. And some genes can cause a very broad spectrum of different clinical presentations. So some genes might cause hereditary spastic paraplegia, but they also might cause other forms of motor neuron degenerative disorder. And recognizing that, we now have a test that covers a broader spectrum of clinical presentations of neurodegenerative, what we call neurodegenerative disorders. An example of that is a gene that Andrew discovered long before I worked with him, a gene called CYP7B1, which we know it causes hereditary spastic paraplegia and is on the gene testing panel for hereditary spastic paraplegia, but it's also on this broader neurodegenerative disorders panel because it can cause a wide spectrum of clinical presentations and that people can have other clinical features. For example, they might have involvement of their vision. So that really backs up our whole hypothesis, which is we need to consider this group of disorders together. So despite the fact that we have identified many genes, more than 76 genes actually, that cause hereditary spastic paraplegia, we still know that we can only provide a genetic diagnosis to about 50% of patients who have hereditary spastic paraplegia. The other 50%, we still haven't identified that gene. That is because there are going to be many more hereditary spastic paraplegia genes to be discovered. What we know is that one of the first genes to be discovered, which is called the SPAS gene, and is the commonest cause of hereditary spastic paraplegia in England or in the UK. It accounts for about 40% of families where hereditary spastic paraplegia can be passed from a parent to a child across multiple generations. The genes that we have discovering today account for a really small number of families. So we've probably got what we call the low-hanging fruit, the common causes of disease, but there is going to be a really large number. We call this the long tail of undiscovered genes, a long tail of genes that account for only a few percent, or even even less than 1% of patients. Andrew will tell you about one of the genes that we have discovered recently, where so far we've only identified 3 families in the world whose HSP is due to that gene. So there will be many, many more genes to discover. And each one will provide answers for families, but it will also give us new information about what underlies that condition, what's going on in the body when those motor neurons aren't working properly. So I'll hand over to Andrew.
Thank you. I start with a horrible looking slide. Anyway, I'm going to try to explain somewhat about the work that Olivia and Nicole and the rest of our team in Exeter have been doing and what we aim to do next. So as Emma mentioned, we know already many genes that can cause HSP, which are linked with all different types of inheritance patterns, and some of them are shown on this. I'm waving my mouse around, but you can't see me waving my mouse around because it's Emma's computer. Sorry, you're going to have to do the mouse waving.
I'm waving them. I was about to start waving my mouse. Don't worry.
So they're shown on this figure on the left, and that's just listing some of those many genes that we know can cause HSP. They're scattered across the genome, and one of the questions is, why are there so many genes that can cause HSP? Do they all have completely unrelated functions, completely unrelated roles inside the cell? Well, that's possible, of course, but it's much more likely, though, that those genes actually work together in a few common mechanisms inside the cell, common pathways inside the cell, and those pathways are the cause of HSP, and it affecting any one of those molecules in that pathway has the same outcome. It impairs that particular pathway and so can trigger HSP. But the main problem that we have had through all of these genetic studies for HSP and other conditions is that, truth be told, for the vast majority of the genes in our genome, even those that we have linked with a genetic condition, for the vast majority of them, we don't really have a very good understanding yet of what their role inside the cell, inside our bodies, actually is. So when we do a genetic study and we identify a gene that causes the condition, if it's a gene whose molecular role isn't well known, then it's not necessarily much help, at least initially anyway. Sorry, Emma, can I have the next slide? Excuse my frog. But on lucky rare occasion, we find a gene that can cause a genetic condition, and the function of that particular gene is really well understood. For whatever reason, it could be in a pathway that's been studied for decades, and that pathway and the role of the pathway in our bodies and our brains in particular, in HSP, is well characterized. And so if we link one of those genes with HSP, then that's really helpful, because that gives us immediately invaluable insight into a pathway that is linked with that condition. So we've been fortunate that we've found 16 HSP genes so far and counting, which are linked with HSP. And from the main part, most of those molecules, the role of those genes isn't well known, but for 2, they had very well defined biological roles. And those those discoveries were really important because they pointed at pathways that we know must be linked with HSP. And these two horrible figures, another horrible figure, my slides are all horrible, so I apologize in advance. These two horrible figures, don't worry about what they're trying to say, but there's the name of those genes are circled in light blue, CYP7B1 and EPT1. They're the genes that we found that have these well understood biological roles, and what these figures are showing you are recipes. They're recipes inside our bodies that our cells in our bodies follow to make fats, to process fats. And they're in two pathways in particular. One is the cholesterol pathway. You'll have heard of cholesterol. Well, cholesterol is a molecule that's processed in a myriad of different ways to make a whole range of cholesterol-related fats. And another pathway, which is called Kennedy pathway, which makes a different group of fats. But the finding of variants in genes in those recipes tell us that those fats, that group of fats is really important for HSP. And so as you'll know, our bodies contain billions of cells, and these are the microscopic building blocks of our bodies. But despite being tiny, any one cell is amazingly intricate and complex. And the one cell is made up of lots and lots of different components, and that's what the horrible figure on the left of this slide is showing us, how complex this, and this is a simplification of what a cell looks like. It's all of the big words which are around the cell label the different bits of the cell, and each of those bits of the cell, each part of the cell, has a specific role inside that cell. And specific parts of the cell are important for those fat recipes, which I just showed you. And we know what those, because those pathways are well understood, we know what those parts of the cell are. And that's what the figure on the right is showing us. That it's two bits of the cell, which are called the mitochondrion, which is the green blob, and the endoplasmic reticulum, which is the purple blob. And they join together. And what happens with, as the cell cooks those fats through that recipe, the various fats in that pathway are passed between those two bits of the cell, are passed between them. And so those two bits of the cell have to join each other and be in contact with each other for that recipe to work properly. And the bit of the cell where they join has got a name, another complex name, which is called the mitochondrial associated membrane. And because I'm not going to say that again, I'm going to use the word MAM for short. So that bit of the cell where those two parts interact with each other, where the recipe flows through, is called the MAM. And so that's important to remember that, because if there's a problem with the MAM, with the way that the MAM is made up, then those two parts of the cell won't work together properly. And so that fat recipe won't work properly. It's like missing out an ingredient or not having an oven to cook a cake. There's a piece of the recipe that's not working properly. So with this information that we now had from finding those genes that were linked with HSP that we know are involved in these fat recipes, we were able to look again at all the other genes that have been linked previously with HSP and see if they might relate to those fat recipes. And that was the hypothesis paper that you'll have heard about, that we've talked about before, that we published, well, actually a year and a half or so ago now, that shows that there is evidence that actually many of these, many of the known HSP genes actually probably do relate to these specific pathways that don't have their own individual function inside their cell, but they are linked with these fat recipes inside of our bodies. I think we can go, yeah, thanks. So that we published that hypothesis paper, which tried to draw attention to these two fat recipes, these two fat pathways, but continued our genetic studies. And shortly after we published that hypothesis, we found our next HSP gene, which was called TMEM63C. We always give genes really uncool names. And what that means, it's a shorthand way of describing what the molecule that the gene makes may look like or may do. So anyway, we found this next HSP gene. This was the three families that Emma mentioned. We found three families in the world that had this form, a recessive form of HSP, excuse me, and some individuals with that condition had mild learning difficulties as well. So we don't have a good understanding yet of what difficulties an individual with TMEM 63C HSP may have, because we only know of those 3 families so far. But what was frustrating was that this was in the we don't know what it does gene pile. So we found the gene, but the role of TMEM 63C, what it does inside the cells, wasn't at all understood. It was a pretty much unstudied molecule. So that meant that we needed to undertake some baseline studies, some basic studies, to try to get some idea of what it may do. And to the chase, what was important and really reassuring for us was that, incredibly, we found that that molecule that is made by TMEM 63C is part of the MAM, the part of the cell that we'd just said will be really important for HSP. And that one tiny part of the cell, TMEM 63C, we found it there. And not only that, we did some other experiments, which I won't explain now, but which showed that its role, we have much to do and much to learn. We don't completely understand its function at all, but everything that we found so far is consistent with it being involved in those fat recipes that we pointed out earlier. So that was very reassuring for us, of course, to see that. All of this work, the left hand of this slide, shows that publication, which has just come out in Brain. And then more recently, sorry, the next slide, thanks. Our newest HSP gene, that the studies are still ongoing, so we've not published yet. And this was a molecule that it is another enzyme, that it is well studied, and it's directly in one of the 2 pathways that we'd already highlighted. So it's specifically in the pathway that we'd said is linked with HSP. So there are now 3 molecules, 3 genes in this pathway. There's quite a few other molecules that are all linked with HSP in this pathway, 2 of which we've found. So again, this was again very reassuring, because having published the hypothesis about these fat recipes, the next 2 molecules that we find are consistent with or directly linked to the pathways that we'd identified. So this was obviously very warming. The next slide, please, Emma. So what about then our future studies? So we're focusing really in 2 main areas. So clearly, genetic studies, we feel actually in many settings genetic studies are somewhat undervalued, but clearly they're crucial. Genes are the keys which unlock the doors to knowledge. So finding genes linked with a disease is really important and really valuable. So we want to continue to pursue genetic studies to find new genes, to understand these pathways and other pathways and other cell mechanisms, and understand how it all fits together better. And the other side of our work is focusing on lipidomics to understand what the outcome is on these recipes, on these fat processing pathways. What does it mean when a gene that causes HSP malfunctions? What does that do to the pathway? How is the pathway impaired? Does it look different? How does it look different? And how does it look compared to other types of HSP? Next slide, please, Emma. So that second line of investigation, the lipidomic studies, is really challenging, and it's challenging because these fat processing pathways are well understood in the order of fats, lipids in those pathways, but it's not understood how different cells, how brain cells, how skin cells, how other cells of our body, how those pathways specifically work in any one cell, and what the amounts of each ingredient in the pathway is. What we do know is that it's really important to understand how the pathways work inside cells, but there's not been the technology really in place previously to do that work, to look at these pathways inside cells properly. So to do that, we've had to develop new methods, and Olivia's work was involved a lot in this to help develop these methods that enable us to understand how these fat recipes, how these lipid recipes inside cells work, what they look like normally, what they look like after you've had a meal, and what they look like, how they differ between men and women, if they differ between men and women, and what they look like in individuals who have a type of HSP. I think then the next slide, please, Emma. And so now that we have the technique to be able to understand, to look at these pathways and look at the fats inside the pathways, we can really, for the first time, understand how fat processing works in HSP using another technique, which is using cell models. So what we do is called the cell model of a condition, and what we do is we use a technique that you've probably heard of called gene editing, CRISPR-Cas9. And what that means is we can take cells in the lab, in the dish, in the culture, in the culture hood. We can grow them up, and then we can target one of those 22,000 genes that we all have. So we would select a gene that we know causes HSP. So TMEM63C, our second newest HSP gene. We've taken that gene and we've targeted it with molecular scissors and cut out the gene. So those cells that are growing in the lab don't have TMEM63C. But the same effect, really, as we'd expect in a patient. That gene with a variant in the patient with TMEM63C HSP, that gene isn't working properly. So the cells of their body can't make that molecule. It's the same effect. We've taken that gene out, so those cells in the lab can't make that molecule. And then we can do what we want with those cells. We can grow them up. We can look at the fat processing pathways. We can do all kinds of studies to understand what the effect of TMEM63C not being there was. And Nicole, who's pictured here, has been doing a lot of this work. She's been generating, alongside the other team members, a number of different genes that we know cause HSP, using CRISPR-Cas9 to take them out and then grow a group of cells that lack that HSP gene. And I think the next slide then follows on from this, which shows that we can use then those new techniques that we've developed to measure those pathways, to measure those recipes inside each type of HSP, and we can compare them together, look to see what things look the same, what things look different, and to show that whether this is a common pathway, I should say, to show that this is a common pathway in HSP. I hope that made some sense. To try to just to round that off. So what we'd like to do is to continue our genetic studies. That's really important, and also to continue our cell line work, the HSP cell lines, to look at how the fat recipes work in each type of HSP, and do a range of other studies to see better how that fits together, to see how the different components of the cell fit together in each type of HSP. Then number 3 on here is a somewhat unusual study, which stems from our work, excuse my phone, sorry, it's my daughter, stems from our work over many years with clinical colleagues who provide care for Amish families with genetic disorders who live in Ohio and Wisconsin in the USA. And in the Amish, because of their culture, certain gene variants linked with a genetic condition can accumulate, can build up. And this can lead to quite a large number of individuals with a particular genetic disorder. So there may be a number of individuals with a condition, but the flip side of that is, because of Amish culture, lots of conditions that are common elsewhere are very rare or even absent in the Amish. So we've identified a gene that's a common cause of HSP globally, which some of you will have heard of, which is the SPAST or SPG4 gene. And that particular gene variant in the Amish has accumulated to a very large frequency. So there are actually a huge number of individuals with that form of SPAST in the community. And it's very likely the largest family described anywhere with that type of HSP, if not the largest HSP family that we know of globally. And so we have plans next year to do a study with the clinical teams who we work with there, who provide care for Amish families, to undertake studies of fat processing in blood. And that will give us, well, because of the numbers of individuals with the same gene alteration, with the same gene variant, it should be a very powerful study, which will hopefully give us much more insights into that particular common form of HSP. The next slide is to say thank you for listening. And that's not me. I do realise we're somewhat similar, but anyway. And the next slide are the individuals who we work with in Exeter, including Nicole. Olivia's not mentioned on here. She's now obviously moved on. And the wider team that we have nationally and internationally who work with us on these studies. So thanks for listening, and let us know if you have any questions.
I think that's wonderful. Thank you very much, both of you.